And here is just a closer look at the the astrocyte, right, and how it's binding to the um, capillary, and you can see these epithelial cells lining the capillary, right, and then these have these tight junctions, meaning nothing can pass in between the cells. All molecules have to pass directly through the epithelial cells, and here we see glucose being brought into the astrocyte, being converted to lactate, and then this can be then given to the nerve cell. Here's a picture of the glutamate neurotransmitter being secreted, and then any excess neurotransmitter can be picked up by the astrocyte and recycled, and then given back to the nerve cell. Here's another look at the oligodendrocyte that has the ability to myelinate multiple axons and multiple portions of the same axon, and this plays a key role in quick transmission of nerve signals for the action potentials. I'd like to show you an EM now of a myelinated neuron. So in the peripheral nervous system, we have Schwann cells that are myelinating axons. So we can tell that this electron micrograph is an electron micrograph of a nerve in the peripheral nervous system, because this is being, the nerve is being surrounded by the Schwann cell, and the Schwann cells are only found in the peripheral nervous system. And this dark region over here, if you look really closely, this is the myelin sheath, and this provides a really great insulation for the axon to be transmitting electrochemical impulses. I would like to point out that unmyelinated axons in the peripheral nervous system also have Schwann cells around them, and you can see the Schwann cells surrounding this unmyelinated axon here and this unmyelinated axon here. However, there is no myelin sheath. So I just want to point out that, yeah, Schwann cells can then still surround the unmyelinated axons in the peripheral nervous system. However, they don't produce myelin. So now I'd like to move on to action potentials. And the resting membrane potential of the nerve cells is kept at minus 70 millivolts. And the reason that this is, these cells have a negative 70 millivolt resting potential is due to, one, the action of sodium-potassium pumps. So I talked about those previously, and sodium-potassium pumps use the energy from ATP hydrolysis to pump sodium and potassium against their concentration gradient. So what happens is the sodium-potassium pumps are pumping sodium out of the cell. So that's why we have more sodium outside of the cell than on the inside of the cell. And then we have more potassium on the inside of the cell than on the outside. So the big K means more sodium, more potassium. Big K means more potassium. The big Na means more sodium. Now, the concentration gradient wants to move sodium inside the cell, and for sodium, and then the potassium concentration gradient wants to move potassium outside the cell. But this imbalance, this concentration gradient, having more sodium on the outside and more potassium on the inside, is produced artificially, or by active transport, I should say, by the sodium-potassium pumps. So and the sodium-potassium pumps are pumping out 3 sodium into the environment, and they're pumping in 2 potassium inside the cell. So pumping out 3 positively charged molecules and pumping 2 in gives the net imbalance, having more positively charged molecules in the outside of the cell than in the inside. A second factor that helps to give the inside of the cell a negative charge compared to the outside of the cell are the fixed anions, such as DNA and proteins that have a negative charge as well. So if you were to take an electrode and put one in the inside of the cell and one in the outside of the cell, we can measure the difference and see that the inside of the cell is going to be negative 70 millivolts, 70 millivolts lower than the outside of the cell. And the way that we can read this is using a machine called the oscilloscope. And these are very useful in studying the nervous system. So the sodium-potassium pump, active transport pump, sets up these two gradients, one for sodium and one for potassium, where there's more sodium on the outside of the cell than on the inside, and there's more potassium on the inside of the cell than the outside. So in order for those two ions to be traveling with their concentration gradient, either in or outside the cell, there are voltage-gated ion channels. So in order for a nerve impulse to begin, sodium is going to have to rush into the cell, and the sodium will be rushing into the cell through a voltage-gated ion channel. So let's take a look at how these work. So here is a closed channel, and they would be closed normally at the resting potential, membrane potential, which would be negative 70, right? So it's closed, no ions can be traveling through. Let's say there's an action potential that's going to depolarize the cell by making it go to a more positive resting membrane potential. Or not resting, but a more positive membrane potential. It won't be resting anymore, but the more positive membrane potential. That would be called the depolarization event. That would trigger the opening of a voltage-gated ion channel, and then ions could go through. After it opens, allowing the concentration gradient to move through the channel, and of course there's not one channel, but there's like millions of channels, right, in the cell, and then the channel will let the ions pass through. Then what happens is there's this little ball and chain that will go and plug the channel. So the channel is going to be stuck closed for a while. And we call this a refractory period, where no more ions can travel through. And then after a refractory period, the ball and chain will release and then let the ion channel return to its closed state. And this is how it is at the negative 70 millivolts. So for an axon to be depolarized, there needs to be a depolarization stimulus. And we're actually going to talk about that towards the end of this video presentation, the depolarization stimulus. And we're going to begin assuming the stimulus has occurred already. And that stimulus, what it does is it actually takes the membrane resting potential of negative 70 and it makes it more positive. So for example, it'll bring it to negative 60 millivolts. This is enough to open these voltage-gated sodium ion channels. So this is going to be negative 70, right? If the if the membrane potential, which is not resting anymore, goes to negative 60, this channel is going to open, okay? And then ions can come through. So I want to show you over here, the resting membrane potential goes from negative 70 to negative 60. The sodium ion channels are going to open, and this is going to greatly increase the membrane potential up to 0. So it's a huge difference, right? And this is going to be so quick. It's going to be in 1 millisecond. It'll jump from negative 60 to 0 millivolts. I want to now jump to the left and show what's happening in this light green area over here, right? So you get the depolarization stimulus. What happened is that the voltage-gated sodium ion channels will open. sodium will begin diffusing into the cell, right? Increasing the membrane potential. And then that'll open other sodium channels, right? Because there's so many of these sodium channels, and if as the voltage, as the membrane potential is increasing, the voltage will change, and then these voltage gated, more voltage gated sodium channels are going to open, allowing this huge increase to 0 millivolts. At that point, we're going to see the membrane potential is going to begin decreasing, and it will actually decrease very rapidly, and even go not only to -70 but surpass that. Okay, and this is going to happen from potassium voltage gated ion channels opening. Okay, so what happens is once this once the membrane potential starts increasing, what's going to happen is the sodium ion channels are going to close, and then the potassium ion channels are going to open. So once we get the proper membrane potential to trigger the potassium channels open, these are voltage regulated channels, they will open. What's going to happen is the potassium is going to leave the cell, and then there'll be less depolarization. Okay? And this, what we're seeing here, this is an action potential, how you get the membrane resting potential to go from -70 to -60 and then to 0, right? And then back down to -70 and even go lower than that to a more negative number. What this picture is depicting is an action potential. So the stimulus really has to hit a threshold, which is -60. So if it just went from like -70, -65, there'll be no action potential. It actually has to go all the way to the threshold of -60 to trigger this action potential event.